So let me start by thanking the organizers, you guys, for choosing our abstract for a short talk and for waiving the registration fee. Always appreciate it very much. And I would like to show you some data of the kidney, where we looked at responses after acute kidney injury, highlighting cell death programs and other interesting patterns, leveraging single-cell RNA-seq. So the rationale for our study was that after an acute insult to the kidney, there is basically two things that can happen to your renal function. One is either it fully recovers, or two is there is one more or less severe way of deterioration of your renal function over time, which we call chronic kidney disease, to which the acute kidney injury then progresses. And that CKD affects 1 in 10 people worldwide, so it's a major health issue. And despite the fact that the kidney has the potential to regenerate, this potential is limited. So what we as nephrologists more oftentimes see is maladaptation and progression towards fibrosis, which is the hallmark of CKD. So the aim of our study was to identify distinct cell types, their transcriptomes and cell composition changes, on the one hand for regeneration and on the other for maladaptation. And we chose a model that allowed us to dose the degree of injury to the kidney, and we chose ischemia reperfusion injury for that purpose, because it allows us to either use short time where we clamp the vessels to the kidney, or a long time, resulting in one, either full regeneration of function, or two, development of chronic kidney disease and fibrosis. And we analyzed 6 versus 6 mice, where we did short versus long IRI, with 3 follow-up time points, harvesting kidneys at 1, 3, and 14 days after ischemia. So leaving us with 2 versus 2 comparisons, and including also 6 control animals that did not undergo ischemia, then all analyzing with single-cell RNA-seq. So these 18 mice then left us with 160,000 kidney cells, which after quality control for excluding ambient RNA or correcting for ambient RNA, excluding doublets, and batch integrating with Liger, left us with pretty distinct clusters of all major endothelial, epithelial, and immune cells, and I'd like to highlight only 2 subsets for the purpose of this talk. The one that we always love to talk about when it comes to injury is the proximal tubular cell, of which there is 2 distinct segments, segment 1 and 3, because it's the most susceptible cell type to ischemia. And the other is the myeloid cell types, namely granulocytes, monocytes, and macrophages. Because when you look at the fraction changes between the groups, the control IRI short and IRI long, you already appreciate that these proximal tubular cells in green are decreased in numbers very, very drastically. And the second thing I would like to highlight here is the drastic increase as we go a long time after ischemia in the long, more so than in the short kidney samples, is how large the percentage gets of these myeloid cell types. So, for example, these granulocytes take up a huge chunk of the cells that we recover in these kidneys. And then looking at these most susceptible proximal tubule cells, when we subcluster those, we get a big blob of mainly healthy differentiated cell clusters with the typical markers that we would expect, and 4 other outlier clusters that are characterized by phenotypes with gene expression such as IL-1 beta, CXCL2, CCL3. So a very inflammatory phenotype, so to speak. And one thing I would like to highlight here is if you look at this orange cluster on the left, that is almost exclusively made up of cells that stem from those kidneys that were harvested 14 days after long ischemia time, so after the very severe injury, meaning that this might be really the cells that classify as maladaptive repair cells. And another way how to look at this is to visualize the cell densities that stem from these certain groups. And you see that in the control and the IRI short groups, the largest chunk of cells comes from the healthy blob, so to speak, whereas in the IRI long samples, the majority is stemming from this outlier inflammatory maladaptive cluster, which is absent in the first two groups, and vice versa. If you look at an example of a healthy subcluster, this is absent mostly in the very severely damaged kidneys. We then went further and did trajectory analysis, asking what kind of lineages do exist in these proximal tubular cells, employing a slingshot and diffusion maps, leaving us with 5 GMM clusters that corresponded basically to 2 lineages. And the first lineage was like this. So when we start at 1 day after injury, down at the bottom of this graph, we then have one lineage that goes along day 3, day 14, and leaves the short 14-day cells right in close proximity to the control cells, meaning they have a signature that is almost like those of the control cells, whereas cells from the very severely damaged kidneys took a different path of maladaptation, and that was more or less recapitulated when we employed RNA velocity analysis and an IRI scoring system that we developed based on the genes differentially expressed between the different groups. We then asked ourselves, what is the gene regulatory network that drives those 2 lineages, inputting the genes differentially expressed between IRI short and long. And this is a scenic analysis showing the regulons that make up, that are specific for these 5 GMM clusters. And again, we had one very outlying cluster that corresponded to the one I showed you before, again, showing that the density of the regulons we found was highest in that cluster. And just to give you some examples, so one example where we'll look at the gene expression of transcription factors that try to define that lineage, that corresponds very well to the regulon activity at the end of lineage 1. And this is HNF4 alpha, TF that is very well known for PT differentiation, so healthy development and meaning regeneration in our context, whereas those other transcription factors were enriched in the second lineage or at the beginning of the damage. And if we, for example, look at this TFC-EBPB and highlight the predicted downstream targets of this transcription factor, we get again those very pro-inflammatory markers such as IL-1 beta and NLRP3 that are highly enriched in this maladaptive lineage number 2. And we then also did enrichment analysis, looking at the different KEGG pathways, for example, where we found that cell death pathways such as ferroptosis were enriched, and in the severely damaged samples, these pathways were sustained, whereas in the short ischemia samples, this was not sustained, but other programs took over, such as fatty acid oxidation or oxidative phosphorylation, meaning that is exactly what a healthy proximal tubular cell would do after it repairs from an acute injury and takes up its normal function again. So when we try to see the enrichment of some of the markers of those necrotic cell death pathways, it was interesting to find that especially markers of pyroptosis, for example, IL-1 beta, and of ferroptosis, this SLC7A11 gene that belongs to a transporter of cysteine, were highly enriched and expressed in this outlier cluster, not so much markers of apoptosis and necroptosis, for example. And lastly, I'd like to show you some cell-cell interaction data looking at ligand receptor analysis. And it was not surprising that the severity and number was increasing with the number of damage. And this is just counting, so to speak, the outgoing and the incoming connections in the proximal tubule. And lastly, I'd like to show you some of those connections when we look at the expression of ligands and receptors of these pro-inflammatory markers. We see that in the control and the IRI short, there is either not much going on, or the signaling is only within the myeloid cell compartment, whereas in the severely damaged samples, we also see expression of ligands or receptors of this pro-inflammatory signaling in PT cells. So meaning we might have a phenotypic switch of an epithelial cell now acquiring an immune cell phenotype. So to summarize, with those 2 degrees of ischemia reperfusion injury, we get 2 different trajectories. One is after short injury, we have a trajectory towards regeneration that is characterized by healthy genes that resemble control cells, that is driven by HNF4A. And when we look at long ischemia, we get a trajectory towards maladaptation that is characterized by inflammasome activation, sustained involvement of regulated cell death pathways, and an epithelial to immune phenotype switch involving IL-1 beta signaling that we're currently following up on in other studies. And I'd like to thank you for your attention and all those people involved in this project, especially my mentor, Katalin Shustak, who's spearheaded this project. And I'm very happy to take any questions.
Thank you very much, Michael. There's a sort of 2 methodological question. Maybe I'll group them. What did you use for the ligand receptor analysis with cell phone DB? And a question about the RNA velocity directionality, where the direction went before the branching, heading towards the origin point. So there's something confusing there.
Yeah, so for the ligand receptor analysis, we use cell phone DB, so it's a created database that most people are using. And for the RNA velocity, it's a question that we get a lot when presenting that data. I started explaining the slingshot trajectory starting at day 1. Because it makes biological sense, whereas in the RNA velocity plots, the arrows indeed were going out from the branching point in the middle. So that would be corresponding to day 3, which from a biological standpoint might also make sense, because that is actually the very diverging point where the 2 lineages decide to diverge either into the maladaptive or the regenerative lineage. So I could try to just define the slingshot trajectory to start at day 3. I think that would not be an issue.
Thank you.
Thanks. So another question from D. Who is, what did you use for ligand receptor analysis? Did you use cell phone DB or?
Yeah.
Else? Okay.
Yeah,
so
I already come. Yeah. Okay. It was cell phone DB.
Yeah. Let me ask another one from Jonas Schulte-Schrepping. How did you subset the PT cells, and did you make sure that this very distinct PT7 cluster is not a contamination of myeloid immune cells or doublets? How many samples did you include per condition? Only 1 per condition and time?
Yeah, that is a very good question. So we subset all samples that we have. So that means 2 biological replicates per time point. So we have 2 animals per short versus long at each time point, and including 6 controls. So that leaves us with 18 animals in total. And in terms of the subsetting and how clean is our PT data, without going back to the first slide, it's in the general UMAP, it's a very, very clean cluster. And we were also concerned about the possibility that it might be contaminated by neutrophils that cling on to PT cells. And we excluded that possibility with employing several doublet removal strategies. And we also validated our findings in an external data set. So we find the same signature of maladaptive repair PT cells in another data set that was independently generated. So we're pretty, and it overlaps very well. And due to time constraints, I didn't have time to show this. And it integrates also very well when we integrate that with our data.
Okay, let's maybe take one more question from John Murray. Very cool talk. The ischemia-specific PT cell cluster was striking. Is this unusual, or do some or most or all other cell types also have these divergent clusters?
Yeah, it would be great to have a look at that. So far, we only looked at PT cells because it's the most important cell type if you look at ischemia. But for sure, I would expect that if you looked at the loop of Henle or the distal tubule, that we would get probably similar responses in terms of the trajectories. But that is that we haven't done yet.
Okay, thank you very much, Michael.
Thank you.